In pharmaceutical manufacturing, the line between a life-saving drug and a toxic contaminant is often measured in micrograms. When a stainless steel reactor is drained, an invisible layer of active chemical residue always remains, trapped in the microscopic abrasions of the metal. For decades, the industry managed this cross-contamination risk using mathematical heuristics. The standard rules dictated that no more than 1/1000th of a normal dose could carry over, or that residue concentrations could not exceed 10 parts per million. This chart contrasts historical compliance with modern risk-based standards. The old methodology relied on arbitrary limits, like 10 parts per million. Now, regulators require limits derived from toxicological data, ensuring residual chemicals pose zero threat to future patients. The old 10 part per million rule failed in two distinct directions. If you were manufacturing a highly potent active pharmaceutical ingredient, a 10-part residue could still cause adverse effects. Conversely, if you were making a benign, low-risk product, cleaning to that exact same limit wasted millions of dollars and thousands of operational hours. A flat numerical limit, applied universally, creates a false sense of security. True manufacturing compliance requires a foundation built on specific, scientifically proven toxicological evidence. This diagram outlines the regulatory framework. At the base, the ICH Q9 standard for quality risk management supports the ASTM E3106 and E3219 structural pillars. They mandate evaluating risk based on scientific knowledge. The core metric enabling this science-based framework is the health-based exposure limit, or HBEL. Instead of asking how clean a surface looks, an HBEL calculates the exact threshold of residue a human can safely ingest. The primary limit is the acceptable daily exposure, or ADE, a specific dose unlikely to cause adverse effects even with daily exposure. This creates a spectrum. Highly toxic compounds sit in the red zone, while low-hazard compounds allow higher limits. To calculate these daily limits accurately, toxicologists rely on NOAEL data. NOAEL stands for the No Observed Adverse Effect Level, the highest tested dose producing no negative physical reactions. This provides a precise safety margin, replacing the outdated practice of extrapolating limits from lethal dose data, which only measured the amount required to cause death. Cleanliness is a mathematically verified patient-specific threshold of safe daily exposure, superseding the simple absence of visible residue. Moving from toxicological theory to operational reality requires looking at how products physically move through a facility. When one batch finishes processing, the equipment must be prepped for a completely different drug. This formula is the engine of that cleaning validation process, the maximum allowable carryover, or MACO. This equation translates the toxicological limits into an exact target for the cleaning crew. The calculation begins with the previous product's acceptable daily exposure. We modify that baseline using the batch size and the maximum daily dose of the next scheduled product. If the next drug uses massive doses, the required MACO limit drops sharply. The decision matrix shows a critical warning here. For high-potency injectables, applying a one-size-fits-all safety factor instead of specific NOAEL data is prohibited. Traditional dose-based math simply cannot guarantee patient safety at those toxicity levels. Because of these variables, MACO is inherently relative. The exact same piece of equipment requires a completely different level of cleanliness, depending exclusively on what product is scheduled to run next. Without the strict application of this MACO calculation, facility managers are flying blind, putting both batch integrity and patient health at severe risk. A commercial manufacturing line can contain miles of piping and thousands of surface square feet. Since swabbing every inch is impossible, engineers must prove the system is clean by targeting the hardest points to wash. This matrix breaks down the 4 specific criteria engineers evaluate to define those worst-case scenarios. You cannot select testing locations randomly. They must be justified across product, equipment, process, and analytical vectors. On the physical side, you identify the product with the highest toxicity and lowest solubility. You then map that against the hardest-to-clean equipment geometries, like dead legs and pipes, complex valves, or rough steel surfaces where insoluble powders catch. On the operational side, you evaluate the process conditions, like the maximum dirty hold time, which is how long residue is allowed to bake onto the steel before washing begins. You weigh this against analytical detectability, prioritizing residues that are difficult for instruments to find. Scientifically justifying where you sample is the only way regulatory bodies will believe your laboratory data on what you sampled. If the location is wrong, the swap result is meaningless. This pyramid illustrates the analytical strategy required once you select a location. Your testing tools must match the calculated risk level. For highly toxic drugs, you need specific methods like HPLC, high-performance liquid chromatography, which physically separates and identifies the exact compound. For routine monitoring of low-risk products, non-specific methods like TOC, or total organic carbon, are faster and perfectly acceptable. To get the residue into those instruments, facilities rely on two primary sampling methodologies: swabbing and rinsing. Swab sampling requires the direct physical scrubbing of an area and is mandatory for targeting insoluble residues trapped in complex gaskets or valves. Rinse sampling is an indirect method used for large enclosed systems, but it requires the residue to be highly soluble so the water can dissolve and carry it out. Relying on rinse water to detect an insoluble compound is a severe failure point. As noted in recent 2024 FDA observations, if the residue won't dissolve, testing the rinse water will yield a perfectly clean result, while toxic sludge remains caked inside the tank. This graphic tracks recent FDA enforcement trends, highlighting exactly these types of failures. The citations overwhelmingly target inadequate sampling plans and unjustified deviations, where facilities change analytical methods without a documented scientific rationale. An elegant mathematical model will instantly collapse under an FDA audit if the physical swabbing and rinsing execution lacks rigorous, documented scientific justification. Scaling this level of scientific rigor across a massive multi-product manufacturing floor requires a system that can manage hundreds of competing variables simultaneously. The Shirakawa matrix plots toxicity against process capability. In the top left, highly toxic compounds in low-capability facilities demand dedicated or single-use equipment. In the bottom right, the variables flip. Benign drug-DEG's highly capable systems might only require rapid visual inspection. However, visual inspection requires mathematically verifying the Visual Detection Index, or VDI. The visual residue limit, where powder becomes visible to the human eye, must be proven lower than the maximum safe surface residue allowed by toxicological data. The Shirakawa matrix provides the operational framework to ensure facilities avoid over-engineering cleaning protocols for safe products while maintaining absolute isolation for dangerous ones. This graphic illustrates the ASTM E3106 lifecycle framework. Validation is no longer a static event tested 3 times and ignored. Stage 3 demands continued verification, using statistical process control to monitor equipment data and detect cleaning drift before failures occur. Transitioning legacy products is a massive logistical challenge, as seen in this Pfizer case study. Every legacy product limit is evaluated. If the new health-based limit is stricter, they implement it and reevaluate cleaning capability. If looser, they retain the older, more conservative standard. The pharmaceutical industry has moved away from the guesswork of flat percentages and arbitrary parts per million targets. The standard is now mathematically proven, toxicologically derived assurance. Ultimately, mastering this complex, risk-based architecture acts as the ultimate shield for patient safety and the bedrock of modern pharmaceutical manufacturing.